So in last class we have discussed up to acute toxicity studies, right? So how to find the low, mid and high doses based upon the different uh, animal weight. Okay. Today we'll see subacute toxicity studies. So it is also known as repeated dose. In this study, they will repeatedly give the doses for the duration of a period like 1 to 3 months, or it will extend to the fourth month also. 1 to 4 months or 1 to 3 months. It depend upon the research. So here they will give the expected therapeutic level. For example, if you got fever means you will go to the hospital, the doctor will prescribe take the paracetamol of specific dose like 650 mg daily, twice daily, or it will the doctors will give based upon the condition. It will the doctors will give 3-day course. So here you are taking the specific dose repeatedly, right? Here they will, after giving the repeated dose, they will check the if any toxicity if any toxicities has found in your body or any complication has found in your body, and everything they will check in the animals in the duration of 90 to 180 days. Okay, so what they will check there, all the parameters, the biochemical parameters, cardiovascular parameters, behavioral changes, if any weight has gained or lost, and everything they will check, the blood test, urine test, and everything they will check here. At the end of the study, they will kill the animal. Why? For the histopathological analysis. Okay, for the histopathological studies, they will kill the animal. After killing the animal, they will take it out the tissue of any organ or brain or heart or any renal, liver. Every organ they will take, and they will find the histopathological changes. If any cells are died or necrosis are happened, if any cells are damaged or not due to the particular dose of repeat, due to the particular repeated dose or not, and everything they will check. Okay, so this is about the subacute toxicity studies. Finally, they used to determine the maximum tolerable dose and nature of the toxicity. Okay, here, so repeated dose studies or subacute studies or subchronic studies. What are the parameters? Check the parameters: behavioral parameters, physiological, biochemical, microscopic observation, histopathological studies, and everything they will check. So the duration of this particular study is 30 to 180 days. So as I told, 1 to 2 months or 1 to 3 months is depending upon the research. Okay, they will take one rodent and one non-rodent species for this particular study. So how they will do this particular study? Based upon the guidelines, OECD guidelines. If you see the 407 guidelines, the procedure will be there for repeated dose of 28-day oral toxicity studies. If you see the 408 test guideline, you will find 90-day oral toxicity studies. That means procedure for the 28 days, procedure for the 90 days. If you see the 410 and 411, it related to the derma. That means if the particular research is going on the to find the skin pharmaceutical products, they will check the guidelines of 410 and 411. Okay, repeated dose oral dermal toxicity, subchronic dermal toxicity for 90 days in the test guideline 411. You can observe the procedures to follow the to do the preclinical studies. If you see the 412 and 413 for the inhalators, what you will take the inhalations, right? Inhalation pharmaceutical products. For that, for in the preclinical studies, they will follow the guidelines of 412 and 413 for the specific days. So that's about the acute toxicities, and we'll move to the chronic toxicity studies. It will be the long duration from 90 days to 1 year or 2 year depending upon the research. Okay, this is a long term effect. They will make a study of long term effect of particular drug. Okay, and they will check here also, they will check lots of parameters like physiological changes, behavior. In every type of toxicity studies, they used to collect the specific data for to get the approval to do the clinical trials. Okay, here at least every 14 days they will make a data of all these things after giving the specific dose. After giving the specific dose, what are the physiological changes has occurred? If any complications are happening into the particular animal or not, behavioral changes. If any blood, they will take the blood test. They will check the WBC, RBC count, platelet counts, and everything. After at the end of this particular study, they will kill the animal and they will do the histological studies. Okay. Why they will kill the animal at the end of the each toxicity studies? Because if you, for example, if you are researching on a cardiovascular system means, so obviously you will check the procedures to do the toxicity studies for the particular animals. After that, if you won't kill the animal for after these particular preclinical studies, continuously if you use that animal into the another research means, that means for example renal system, that means it won't give the accurate results over there. It makes a lots of complications to that particular animal. That's why each research we have to, after end of this particular study, we have to kill the animal for the histological studies. Okay, each and every animal they have to kill and they'll check the histological changes, either sex, male or female, and everything. Okay, so that's about the chronic toxicity studies. So here check the objective of chronic toxicity. So why we have to do the chronic toxicity studies? To evaluate the cumulative toxicity of chemicals, and you also assess the carcinogenic substances in the particular research product. It is very important to find the carcinogenic substance which is present in the research product. Okay, so the particular duration depends upon the rodents, means 6 to 24 months, and non-rodents 12 months or longer than that. In these toxicity studies, they will utilize the two species, rodents and non-rodents, and they will check the parameters like mortality, pathology, and histopathology, weight changes, and clinical pathology of animals. They also make a data of particular death report also. Chronic toxicity. So we are finding the toxicity studies. We are finding the particular research product, it is making complications to the animals or not, everything we have to report. So that's why they have to make the mortality rate, and they have to check the pathological changes after giving a particular dose of to the animals, what the changes they will occur in the body. It is getting making complications to the body or well and healthy of animals, the behavior after this at the end of this particular study, how to make the histopathological changes also to find the cell damage or any cell damage or necrosis happens or not. We have to check for each and every organ. We have to check consecutively 14 days the weight change, if any weight is gaining or loss, and we have to find out the factor of weight also. If gained means how it is gaining, due to the particular drug, or we have to relate with the drug itself. If a particular rat is lost weight means we have to check if any complications happen in your body, means we have to check everything. Okay, every report we have to make chronic toxicity. So let's see the overview of animal toxicity studies durations. So acute toxicity they will make a duration up to 14 days. Subacute repeated doses I told right 28 days. Subchronic 3 months. Chronic toxicity study 6 months to 2 years. Okay. So these are some regulatory mechanisms in India. That means we'll get approvals. So I told that after making the preclinical studies, for doing the preclinical studies, we have to take approvals from these regulatories. So Institutional Animal Ethicals Committee, CPCSEA, Committee for the Purpose of Control and Supervision of Experiments in Animals. That means um, I think the cultivation of animals, the usage of animals in the laboratories, the laboratory conditions, all the procedures, which is the situation, which is told in the CPCSEA guidelines. Okay, these are specific regulatory bodies. We have to take the permissions before doing the preclinical studies. See, Prevention of Cruelty to Animals Act, if we don't take the permission from these regulatory bodies, that is crime because we are killing the animals unnecessarily. So we have to take permissions. They will suggest you if you are for the acute toxic studies. If you want to do 100 animals, means that is offense. Okay, we don't need 100 animals for only acute toxicity studies, right? For acute toxicity studies, we need only 4 batches, 3 in a batch. 12 animals is needed, that's it. Why you are killing unnecessary more than 50 animals or 60 animals? So they will suggest the committee has made a procedure um, for to give the approval for to do the preclinical studies. First, you have to give the plan to the particular protocol. We have to give the protocol to the higher authorities or regulatory bodies. Then they will check the protocol and they will give the approval to do the preclinical studies. Okay, so they used to do in vivo studies and in vitro studies. So in vivo studies means they will a pharmacological study should be designed. It means inside your body they will check the dose response relationship, physiological changes inside. In vitro means they will take any part of the body and they will check the dose response relationship. Okay, they'll check the dose concentrations. For example, they'll take a rat abdominal tissue from that. For example, they want to do research on acetylcholine means acetylcholine is one of the drug. Okay, so acetylcholine, they will check the dose concentration, they'll increase the dose, it is responding to the tissues or not, and everything they will check. Okay, in vitro studies means doing outside. In vivo means inside your body. Okay. So types of local toxicity studies. So depending upon the toxicity studies, they will choose the specific animal for the accurate results. So for example, a dermal toxicity studies, they will utilize the rats and rabbit. If you see the dermal phototoxicity studies, they will check on the guinea pigs. Okay, vaginal toxic studies, they will check on rabbit or dog. See, depending upon the variation of particular toxicity studies, they will choose specific animals to do the preclinical studies. So I have make a list, check it out this thing. See, so for the parenteral drugs, have everything for inhalation, one rodent and one non-rodent species required specifically for acute oral toxicity studies, ocular toxic studies, albino rat is required. Okay. So I told that OECD guidelines for based upon these test guidelines, we have to do the procedures in the preclinical studies. So if you by seeing the headlines, you can check the guidelines. So acute toxic studies 401, acute oral toxicity 402, acute dermal toxicity Subchronic 407, 408, 409, 410, 411. See, chronic 452, 453. So OECD guidelines are full of stuff is there. So if you want to do chronic toxic studies, directly move to the 452 guideline and check the procedure. Okay. So chronic studies. So make maximum tolerated dose and highest dose tested. That means, see, maximum tolerated dose is the highest dose that can be tolerated without significant lethality from causes other than tumors. The particular animal can tolerate the highest dose. So what we have seen in the acute toxic studies to finding the low, mid and high. Okay, depending upon the particular plant extract what we are collected from the plants and for utilizing for the research purpose, that extract they have to find out, right, what is the highest tolerable dose and what is the highest toxicity dose. Okay, lethality means killing. Okay, up for some extracts they will fix up to the 2000 milligrams per kg body, but that is the maximum tolerable dose that we can give to the animals. What is the highest dose? Highest dose that can be expected to yield results relevant to the humans. So why we are doing the preclinical studies? For the safe studies we have to do in the clinical studies for the safe to the humans, right? That's why they will check the toxicity studies for the particular research product in preclinical studies. Okay, that's why they will do toxic studies in animals. So we have to know some two terminologies like ED50, effective dose 50, and lethal dose 50. So effective dose 50 is nothing but the medium effective dose is equal to the dose for which 50% of animals exhibit an effect and half of animals exhibit no effect. Okay, ED50, effective dose, therapeutic dose. It is effective to the particular animals. So in one group they will put 30 animals or 25 animals or 20 animals depending upon the particular procedure. For example, if you take 20 animals in a group, means if you are giving effective dose of your particular research product, 50% of population, that means 10 animals, it is getting effect, therapeutic effect, it is getting cured from the particular diseases. Okay, that is nothing but effective dose. Lethal dose means the effective may defined as the specific toxic event or sometimes defined as lethality. Lethal dose, 50% of population as killed or maybe complicated happens to the particular animals. So in a group if you take 20 animals, 10 animals due to this particular dose, 10 animals has died. So that is nothing but the lethal dose. Like that, effective dose 10 and effective dose 90, which 10%, 90% of animals respectively demonstrated effect. Okay. So you have to know about another terminology is limit dose. A dose which is considered high enough that if no mortality or significant toxicity is seen in animals receiving this dose, no higher dose are required. So by doing the research in the preclinical studies, they will find the limit dose also. Okay. So for example, they have mentioned here, see, acute oral toxicity studies 5000 milligrams per kg body weight is the limit dose. For acute dermal toxicity 2000 milligrams per kg body, that is the limit dose that we can do in the preclinical studies. Okay. So I told that they will collect the parameters in the toxicity studies, all acute, subacute, and chronic toxicity studies. They'll collect some parameters. For example, convulsions after administration of specific dose. Is that animal get convulsed? Fits may happen, seizures happened or not, and everything they will check. If any involuntary tremors happen or not, everything they will make a note. Okay, so involuntary actions, involuntary movements, they will make a collection specifically the convulsions happens or not, and they will check the ocular signs. Okay, in the eye, if any conjunctivitis or corneal opacity, iritis, the inflammation of iris, inflammation of conjunctive, specifically they will check each and every minor part, tiny parts also. Okay, so cardiovascular signs is any tachycardia or bradycardia, any vasodilations and vasoconstrictions, arrhythmias, is every part, every science they will collect and make a data, because those are all we can see in the humans, right? So that's why if you're taking this drug, for example, if you're taking for head pain reliever, if you're getting some stomach ache or if you're getting some heart rate increased, means that is complication that we have to check. That's why we have to check the toxicity studies in preclinicals. Okay, that's why they will check the toxicity studies of specific drug. Okay, if you are taking a drug, means it should be act on that system only. It won't involve in other systems. That makes the complications. Okay, so that's why they will do the toxicity studies. Autonomic science. If any complications happens in the autonomous nervous system, everything they will check, sympathetic system and parasympathetic nervous systems. They'll make a data of specific animal. So other signs like salivation is piloerections, analgesia, muscle tones, muscles are correcting workly or not, the nervous system, the neurotransmitters are giving signal to the muscle or not for muscle contraction or relaxation, and everything they will check. These are the specific parameters they will make a data for each and every animal, each and every group. So those are the specific parameters make in the collect the data in the preclinical studies. So after preclinical studies, they will produce all these data to the regulatory bodies, higher authorities. Then they will check the each and every day report, and after making this after collecting, after recorded this data, then they will give approval to do clinical trials. Okay, if they fail in preclinical studies, means again they have to come from first step. Okay, then after preclinical studies, they will move to the clinical trials. So in phase 1, in clinical trials you will find 4 phases like phase 1, phase 2, phase 3 and phase 4. If you see phase 1, only 20 to 80 population will be that healthy volunteers they will check. Healthy volunteers. After getting approval of phase 1, they will move to the phase 2, 100 to 300 participants of patients. In phase 2 and phase 3, they will check on the specific patients. If you are doing a research on particular renal system, means they will collect the renal system patients of specific diseases, and they will give that particular research product to those patients. Phase 2 and phase 3. Okay, if you see the phase 2, 100 to 300 participant, if you see the phase 3 increasing in number 1000 to 3000. So if you got successful up to phase 3, means they will report that particular data to the higher authorities and regulatory bodies, and they will get approval. It is safe. So till phase 3 we have made a research. Till now no complications has happened. Any other complications happen, if you will find some adverse effects, those are minor adverse effects, those are acceptable. After that they have to take a approval from FDA, Food and Drug Administration. They will make a review from preclinical to clinical phases up to phase 3. Then after getting approval, they will move to the phase 4, post marketing phase. Here in this phase, the drug will come to the market. Everyone, more than 1,000 people, they will take that particular drug. After getting drug approval, it will come to the market. So it will roam around the world. Then they will make a monitor of the particular drug. That is the post marketing phase. They will make a monitor of particular drug. If any complications happening to the particular environment, depending upon the environment, the drug stability may vary. Okay, for that purpose they will uh, make an monitor of particular drug phase 4. For example, uh, in some pharma news you have seen about this particular drug is banned, no more usage of this particular narcotic drug. That means they are making monitor of particular drug in the post marketing phase. Okay, if lots of complaints has formed against the particular drug, means after making the collection of particular data, the complaints report and everything, then they will ban the specific drug. Okay, in India we are not having specific system for that. If you see the foreign, for example, I told that example, if you're taking for a head pain reliever, the particular drug, it is relieving the head, it is also making trouble in the stomach. That means that is complication, that one of the adverse effects side effect is doing. So they will complain, they will raise a complaint against a particular drug and the company. So if lots of complaints has come from that particular drug, means, and they will check the patient's reports and everything, and they will ban the specific drug. So that is about the phase 4. So that is about today's. Thank you. Have a nice time.